Cool. All right. So let's now go on to the final task of the week 6, and that's looking at how meiosis increases variation. We've already talked about one way it does that, and that is through the fact that we're halving the chromosome number, and we're getting the mixture of both the male and female genetic material. You have 2 parents, but there's actually more layers to how there's more layers to how we can get even more variation. This one's going to be a big kind of thing to think about. Obviously, we're still working at number 7, which is that greater variation than asexual reproduction. But we're also going to learn more reasons to why. So we're looking at the methods of how meiosis can do that through independent assortment, crossing over, random fertilization. We actually talked about, not really random fertilization. We haven't talked about that yet. We've talked about the 2-parent thing. Cool. And the other thing we're going to talk about is how crossing over can result in new recombinations of chromosomes. So with meiosis, there's 3 main things we're going to be adding to our understanding on how it increases variation. The first is independent assortment, then there's crossing over, and then there's random fertilization. So in order to kind of introduce some of these ideas, what I have is some really cool real-life examples. So in this case, we have 2 girls, and I like to ask the class if they think that these 2 girls are friends, cousins, or sisters. So take a moment and have a little guess, and I'll give you the answer. They are actually sisters, and not only are they sisters, they are twins. They're not identical twins, they're fraternal twins, and I think it's really cool to kind of realize in the same family you can get 2 very different traits. So let's think about the why and how that is possible through science. So the way this happens is through something called independent assortment. So if you remember back when we were talking about meiosis, we had the homologous chromosomes pair up. Now this pairing happens in a random way. And obviously remember we have half the genetic material from mom, half from dad. And either one can line up at the equator or at the center of the cell. So if we go to the notes that I wrote in the last lesson, I just happened to draw it so that way the red chromosomes were on the same side as the blue chromosomes. But it doesn't need to go in that way. It can also go in, this orientation where I get a red and a blue and then a blue and a red. So that's where it's going to divide down. So both of those are possible. So each homologous carrier of chromosomes will segregate itself independently, thus the term independent assortment. So that means that there's a large range of possible combinations because you have to remember there are 23 pairs of chromosomes. So since we have 23 pairs, each one's going to line up on its own without any influence to the other ones. And so what this means is that if I do 23 to the power of 2, because each one acts independently, I can get a possibility of 8 million different combinations when it comes to creating the egg and the sperm, which is really, really cool to think about. So this is the first layer of variation that we're getting, and this is kind of showing the importance of having those 2 parents. So let's go back to my 23andMe. So in my case, I have really, like I said, organized chromosomes because I have, my Asian mom and my white dad. When my body has produced its eggs, there's going to be random combinations of, do I get mom's chromosome or do I get dad's chromosome in each one of those eggs? So I'm going to have some eggs that are going to have a mixture, some that might by chance have more Asian chromosomes, some by chance that might have more of the European chromosomes. So my eggs are going to look completely random, and it'll be really interesting if I have kids and I can get them a 23andMe as well, because then I can see what their chromosomes are going to look like. So that's kind of just neat to think about. The second layer then of variation is that random fertilization. So basically, it's that randomness of whatever egg gets released and whatever sperm that meets it. When that fertilization occurs, it makes a zygote. So that's that single cell post-fertilization beginning of an organism. And then once you have that fertilization, it's going to start doing that mitosis to create more cells to make that organism. And you can see that in that newt or tadpole video that I posted. So with random fertilization, the process is random. So there could be any combination of sperm and egg. And that gives, again, that huge variety of that possible combination. That's why siblings don't necessarily look like each other, even if they have the same biological parents. So thinking about how this was possible with the twins is that both of their parents were biracial. So they were both black and white. And by chance, through independent assortment, the mom created an egg that had mostly white alleles. And so I say white alleles as in like the European traits, and released another egg that had typically, black alleles. And then by chance, the sperm that fertilized it were the same as well. So the dad is also biracial. And so the offspring then just happened to get all of the European genes and all the African genes. And that's how you have these twins that look different to each other, even though they have the same parents. And what was also interesting about this family is that they had another set of twins, and that happened again. So really kind of random and insanely lucky, because this is very, very rare for something like this to happen. Cool. So I'm trying to think if there's anything else I wanted to show you with that 23andMe. No, I think that's it. So let's talk about the next way of variation. So obviously, one chromosome can have multiple different genes on there, and the genes that are on the same chromosome are known as linked genes. So that means that when they're linked, they're often inherited together. And we can see that in the general population as well, because you notice certain traits get inherited together. So for example, dark eyes, dark hair, that tends to get inherited together. Light hair, light eyes tend to get inherited together. However, we know that other possible combinations can happen as well. We can see people with dark hair and blue eyes. We can see people with blonde hair and brown eyes. We can also see, the darker complexion with those blue eyes. So I just wanted to show you guys just the range in genetic variation. So even if genes are linked together on the same chromosome, we can actually get another shuffling around of the traits. And the reason why has to do with something called crossing over. So when the homologous pairs line up in that first meiosis, in those homologous pairs, what they're going to do is swap some of their DNA around, and they shuffle it. And what this is doing is now we are creating 4 different sister chromatids. They're not identical. So again, we get new variations. So genes that were previously tied together are now no longer tied together. So this is known as recombination. So in reality, when you're looking at your chromosomes, it's not only a blend of the independent assortment, but it's also the independent assortment on top of the crossing over with the random fertilization. This is really easy to kind of show with Play-Doh. So hopefully when we're in person, I can get you to play with some Play-Doh and actually kind of do this. This is also, again, like I said, a source of potential mutations. Sometimes the wrong amount of DNA gets transferred over, and one sister chromatid gets shorter and the other one's too long. It could also result in that inverse of the DNA as well. So there is potential risk to it, but it's worthwhile because you get that that recombination of genes. That's just another one of my jokes. Cool. So that's all for that lesson. But before we move on, we'll go back to the 23andMe. So right now, my chromosomes, like I said, are very clean cut, half from mom, half from dad. They're going to randomly arrange, but on top of that, they're also going to recombine and shuffle the genes around. So if I have kids and I get them a 23andMe test, I would expect their chromosomes to actually have a mixture of the blue and the orange, or the blue and the red, because of the recombination process that has occurred in that random crossing over. We do see this little bit of red for some reason in like my European chromosome. I'm not really sure what that is. They're still collecting data, obviously, with these chromosomes and 23andMe, and there's obviously stuff that they don't know yet. But yeah, so it'll be interesting to see a DNA profile of my potential offspring, because they're obviously going to look very different. And also that's going to influence then what traits they get. So, like I said, it makes the twins even more amazing, because whatever crossing over had happened, it didn't actually change the physical features too much, and then they randomly assorted, so that way they inherited most of the European traits and then most of the African traits. So yeah, I just think it's really neat. Anyway, that is how meiosis creates variation, and we know that variation is going to increase the chance of survival with a species. And you should now know independent assortment, crossing over, and random fertilization. You should be able to explain them and list them as sources of variation. And you should also explain how that crossing over results in that recombination. We will look at this some more when we start doing those larger Punnett squares as well. So now it's time for your mahi choice. Go to the Google site to find items to work on. Remember, I don't expect you to do everything. It's just to give you some differentiation and some choice. And all my resources are on the Google Classroom.